Screen is visible?
Yes, ma'am.
Yes, ma'am.
Okay. So today's class is on gene expression studies, where we will learn many things. One means everything related to RNA, with which is first that if you have to detect a particular transcript, then which are the various ways you can detect it. I had briefly mentioned that you can use northern blotting, but then there are other methods as well. If you want to know that for a particular transcript, if there are alternatively spliced forms also, then can you detect those? Then if you are focusing on multiple transcripts in the whole cell, I mean, you are not concerned about only one transcript, but you want to measure the changes in the RNA level at the whole transcriptome level, then which technique you may use. If for a particular transcript you want to know what are the ends of a particular cDNA, to exactly map what is the transcription start site and what is the end site, then which are the various methods. And finally, if you want to study its promoter, you want to know that how a particular gene is getting expressed or getting affected by the transcription factors which are binding to the promoter, then which are the various methods which are concerned with it. So this whole thing is, there is a chapter in TA Brown also, and some extra notes are given as part of this particular lecture also. So when we talk about RNA, and we have to work with RNA, obviously the first step is RNA isolation, and everything is same like you do for DNA isolation, which is that you first lyse the cells, and then you have to remove all the debris. You have to remove the proteins. So what you do is you give it a phenol chloroform treatment, right? And like we used to do it for DNA isolation. And what happens that RNA comes in the this soluble form, and then there is organic phase, and there is a middle phase where these denatured proteins are there. The only difference is that here we keep the pH of the phenol in the range of 4 to 5, around 4.5, because RNA is soluble and comes in the aqueous solution when we use this low pH. While DNA, it gets denatured and it is present in that middle, that white precipitate where proteins were also present. So this is the way you can specifically isolate RNA, leaving DNA. So this is the major difference between the two, but otherwise the points are same. You first have to lyse the cells, you have to take care that no proteins are there, so you give phenol chloroform treatment. To ensure that DNA is not there, you reduce the pH while you are isolating RNA, and so that DNA doesn't come into picture. Once you have RNA into solution, then you have to precipitate it. How do you precipitate it? You can use isopropanol, equal volume of isopropanol, or if you remember, you can use 2 volumes of this ethanol along with 3 molar sodium acetate. So it precipitates any nucleic acid. So those two methods you can use. Alternatively, the aqueous phase that you have got, you can pass it through the spin column, and the RNA will bind to silica membrane, and then you can elute it out. And like for DNA or for RNA, you have to give 70% ethanol washes. Why you have to give? Because you want to remove the salt from your nucleic acid. So you treat it, you means clean the, you remove the salts by 70% ethanol washes, in which the 30% of the water takes away the any salt which is present, while 70% ensures that RNA remains in the precipitated form. And then you will dry your RNA, and then you will dissolve your RNA. Once you get your RNA, then you will take its OD. So, like in case of DNA, if you remember, 1 OD was equivalent to 50 microgram of DNA. And in case of RNA, 1 OD was equivalent to 40. So these are the major differences. Like for DNA, you will again look for 260 by 280 ratios and so on. Another way to look for the quality of the RNA. See, one is that measuring that what is the amount of RNA that you have got. Another thing is that how do you know that what is the quality of your RNA? So, like in case of DNA, also you run it on gel and then you see that okay, whether my DNA is good quality, whether yeah, I'm getting in case of plasmid, obviously you get 2-3 bands, but if it is you are getting a very strong supercoid band, then it means that your plasmid DNA quality is very good. Or let's say if you are isolating genomic DNA, again you will see that it should be a very very high molecular weight band that you are getting, and with minimal smear in the solution, in the well that you have loaded it. Similarly for RNA gel, you will run a gel. The only difference is that the gel that you run, it is a denaturing gel. It has formamide and it is run in the presence of formaldehyde. So why do you need a denaturing gel for RNA? Because RNA is single stranded, and because it is single stranded, it tends to form various secondary structures. So, when you want to run it, you want to get away with the secondary structures. So, that's why you run it on a denaturing gel. In fact, even loading means before loading, the RNA is given in some premix buffer. It is treated at 65 degree for I remember like 10 minutes, and then it is run on gel, so that you don't have any secondary structure and you get beautiful picture of RNA. So, how will your RNA look like on gel? So, if you remember that RNA belongs to various categories within a cell. You have the most prominent of which is rRNA. So, it is like 90, more than 90% of the RNA is rRNA only. So, it is only 5% that you have mRNA in the means if you want to categorize on the basis of percentage of RNA, then mRNA is just 5%. So that's why when you run it on gel, you see two prominent bands of rRNA, which in case of eukaryotes, it will be 28 and 18S, while in case of prokaryotes, it will be 23 and 16S, right? And then you will see this background, very light smear will be there. What is that smear for? It is mRNA. So why do we see a whole smear type of thing from up to down? Because obviously we know that genes are of different size, and accordingly the cDNAs are of various sizes, and mRNAs of various sizes. And when you run it on gel, you will have some mRNAs which are very high, which have a very big size, while some mRNAs will be very, very small. And also, maybe towards the end, we also see microRNA and other small RNAs which are present at the base. So that's why we get a whole range of this background, very light smear at the back. So when I run it on gel, and if I'm able to see these two strong bands and a very light smear at the back, I'm sure that my RNA quality is good. But if I'm not able to see these two bands, I'm sure that my RNA is heavily degraded. If means even more degraded would be that you find everything at the base. So it means that your RNA is completely degraded and you have to isolate it again. So again, if I compare DNA isolation versus RNA isolation, I want to tell you that for RNA isolation, you have to take utmost precautions because RNA has that OH group, 2' OH group, which is more prone for hydrolytic attack. So it is much more prone for degradation. So when you isolate RNA at various steps, there is you may also want to add RNase inhibitor, and also it is prescribed that you don't speak because RNAs are everywhere. So you have to be you have to do it in very clean environment. You should not speak over your tube when it is open, so that just in case RNAs can degrade your RNA. So all these precautions you will see in various these RNA isolation protocols, that you have to take utmost precaution when you are dealing with RNA, be it for isolation or be it for further any use that you are using it for. Now, one way is that you run it on gel, but now a more sophisticated systems have come. which are chip based, and in which what you do is that you isolate your RNA, multiple samples, and you run it. I mean, you load it on a small chip. This instrument is called bioanalyzer. Our department, I think, as of now, we don't have it, but KSBS does have one bioanalyzer. So, and then you just wait, means the output comes in the form of this graph, where these two peaks, they represent the 28 and 18S, or 23 and 16S, whichever organism RNA you are isolating. And if these peaks are very strong, then you are sure that your RNA quality is good. And it also gives this picture as part of bioanalyzer analysis. So I'm not going to detail of it, but basically, yes, there are other ways to look at the quality of your RNA. And many companies, when we send our samples, let's say for RNA sequencing or for microarray analysis, they first run your RNA on the bioanalyzer and then say that your RNA quality is good. So we will proceed with the next steps. Otherwise, they will just say that your RNA quality was not good and we are returning your samples, or we will not proceed. So, yeah, just a brief introduction to bioanalyzer. Then comes, so what we have done till now, we have isolated RNA. I've told you some precautions that you must take when you are dealing with RNA. How do you check for its quality using any of these analysis? And also that the quantitation, the only difference is that for DNA it was into 50 and here it is into 40. Then comes, if you want to detect a particular transcript, you are interested to know that, let's say, the gene I'm interested in this case, let's say there is a gene called BAG1. What are its levels in normal cells versus cancer cells? So, how can I do that? So, what I will do, I will isolate total RNA from normal and total RNA from cancer cells, and I will run it on gel. So this whole technique that I'm talking about is northern blotting, and it is exactly similar like southern blotting, only with the change that in this case you will isolate total RNA and run it on gel, while in case of southern blotting, which I told you in my last class, it was the DNA that you run on gel. That's the major difference. And obviously, I told you, because you have to run RNA in denaturing conditions, so the running gel and the buffer are different as compared to DNA. So once you run it on gel, then you transfer it to a membrane. How do you transfer it? It's exactly like southern blotting. So I'm not going through. If you remember, there was a wick and there was this capillary action by which the DNA from the gel is then transferred to a membrane. Once it is transferred to a membrane, then you do the hybridization with a probe which is specific for these gene or for a housekeeping gene. So, what is a probe? That you must have already been told in your assignment or practical session. So, these are oligonucleotides which are specific for your particular gene of interest. So, why they are called probes? Because they are generally labeled. They can be radioactively labeled or they can be fluorescently labeled. So, then what you do is that the membrane that you have got, you incubate that with a solution in which you add your specific probe. So what will happen that among this various RNA which are present on this membrane, your probe will go and bind to your gene of interest. And then when it binds, and we take care that this probe binds very stringently only to your gene and not to other uh, RNA which are present in this, by giving a very highly stringent conditions and washes. And then you expose your membrane to a X-ray film, and what we will get are these black bands, and on the basis of which we can come to a particular conclusion. Now let's say ignore the lower part. Okay, so what I say see is that I see very light bands here and some dark bands here, and maybe from this particular figure I can completely I can make a lot of presumptions. One, that indeed my gene of interest is highly expressed in most of the cancer cell lines as compared to the normal cells. This conclusion I can make. But anybody can argue that it could be that Ritu, probably you did not load enough RNA here. Maybe you loaded very less RNA here and you loaded more RNA here. So how do I convince those people that no, I did not do that? For that, I have to also do probing with a normalization control. So what are normalization control? Normalization controls are, let's say in this case, the probing done for a housekeeping gene. So what are housekeeping genes? Housekeeping genes are those genes which are expressed at very high levels at any stage of the cell or under various conditions. So we believe that the expression of housekeeping genes generally do not change in any of the conditions that we generally talk about, be it normal versus cancer, be it heat shock versus control, means in response to any hormone treatment or transcription factor treatment. So we believe that the expression of the beta, in this case, beta actin is the housekeeping gene that we have taken, it remains same throughout. So if the level of these are same, then I can convince that, see, I loaded same amount of RNA in these samples and same amount of RNA, let's say in all these samples. And that's why I can make the conclusion that as compared to normal, in majority of my cancer cell lines, my gene of interest, BAG1, is highly expressed. Of course, I can also definitely conclude that in this particular cell line, even though my beta actin gene was high, I mean, it is almost similar or rather little bit high. It could be that I loaded little bit more RNA here, but still my gene of interest is not highly expressed. Definitely it is lower as compared to this or almost equal. So these type of conclusions I can make, but at the same time I can also conclude that okay, in all these other ones this is definitely highly expressed. Another thing is that if you just see these two samples, I'm just giving you various scenarios. So maybe if I just see this particular this photo, then I can say that probably my in this particular cell line, my gene is highly expressed. But if I look at my beta actin, which is representative of the total RNA that I've loaded, I see that actually here I loaded more RNA. So when I normalize my samples on basis of this, then I cannot conclude that this gene is highly expressed in this particular sample. So that's why normalization control, be it for northern blotting or be it for real-time PCR experiments, which I'll just come to explain. RT-PCR experiments, they become very, very important. Now, as you can understand, this is a more laborious process because you have to run it on gel. You have to wait for several hours for it to be transferred to a membrane. Then you have to incubate it with a probe. First, you have to make a probe. Then you have to incubate it with the probe for a few hours. Then you have to give washes, minimum 3 to 4 washes to remove off the weakly bound probe, and then you expose it to a membrane, then do developing, then do exposing, etc. So obviously it is a much time consuming process, and do we have an alternative for that? And also, as you can see, it also relies on some in this case radioactivity, and I told you in my last class how difficult it is to work with radioactivity. So yes, we do have an alternative option to, if you want to check the level of a particular transcript in various tissues, for example, in a particular plant, then you can use a technique called RT-PCR, which is that you design primers which are specific for your particular gene of interest. For example, I can design primers which are specific for BAG1, How do you design primers? We have already taught you in assignments. And then you can detect your gene of interest. So the only difference is that before PCR, because we want that the amplicon should only come from RNA and not from DNA. So one thing is that you have to design your primers mostly from intron exon boundaries, and also it will require a pre-step because this PCR cannot be done using RNA as the template. You have to first convert your RNA to cDNA. So first step is you do reverse transcription, which is done through these various reverse transcriptase, MMU, LB, reverse transcriptase, AMB. So these were some examples I told you during the start of my this whole course, which enzymes may be used for what purpose. So anyway, you make your cDNA, you remove the RNA through RNase removal, and then you use this as a template for performing PCR. And these primers, how do you design it, will be very important, because on basis of this only you have to come to a conclusion. If your primers are binding to multiple regions in the whole genome, I mean to multiple transcripts, obviously you cannot figure out that the various multiple bands which are coming when you're running it on gel, they are coming from which particular gene. So that's why this designing is very important, that it only binds to your gene of interest. And here again, this is actin. So this is the housekeeping gene. You see that irrespective of which plant organ I've taken, its level remains same, which suggests that, okay, I used same amount of RNA in each of these cases. And despite me using same amount of RNA in all these cases, I can conclude that this whatever this gene is, KNOX1, it is very highly expressed in apical meristem, while in leaf it almost has zero expression, right? And similarly, it's another maybe it's a gene family, I've just taken it from Google just for representative purpose, and knox2 gene will probably belonging to the same family. It is highly expressed in ovary as compared to the knox1 gene. So means these conclusions you can draw, and if you want to know the transcript level for a particular gene that you are interested in. But what about those cases where I want to know, I'm not restricted to knowing the transcript level for any 1 or 5 or 10 genes, which I can easily do by designing primers against those, let's say, 10 genes. But what about if I'm interested in those 25,000 different genes which are present in the whole organism, in the whole cell, which means the whole, so every cell will have a particular number of transcripts here and different type of transcripts and so on. And if I want to know the expression level changes of all the transcripts at the same time, then one technique is microarray. It is still a very prevalent technique, very widely used technique. And here what is done is that it is based on a chip. So on a chip, what they do is that they spot the oligonucleotides which are specific to each and every gene which is present in the genome. Okay, so means they are complementary to it. So on this spot, let's say I have oligonucleotides against BAG1 gene. Here I have oligonucleotide against actin gene, here against myosin gene, and so on. So every spot is a separate gene, and sometimes they put multiple spots for the same gene, I mean just for as a replicate, as a surety that your gene is actually showing similar things. So replicates for a same gene are also present. Okay, so how it is done is that, for example, you have a normal cell and you have a cancer cell, and you isolate total RNA from it and then isolate further mRNA from it. So that can be or you deplete your RNA by rRNA removal kit, and you just isolate mRNA. So then what you do is you make the cDNA, and when you're making cDNA, you just ensure that the cDNA, in this case, it is labeled with, let's say, green fluorescent probe. And here, when you make the cDNA, it is labeled with, let's say, a red fluorescent probe. So what we have done, that all the RNA which will come from, let's say, cancer cell will be red colored, and all the RNA which is now represented by cDNA, it is now green color from the normal cell. So now, when what I will do, I will combine these probes, these two things, and then I will hybridize it to the chip. So what will happen, that I will get different colors at various places. So let's understand that what do these colors represent. So the well, which does not show any fluorescent, it is just black, still black in color, it doesn't show any fluorescence, is it is representative of those genes which is present in the genome but is not expressed in that particular tissue that we are talking about, or that particular cell that we are talking about. So there is no expression of that particular transcript. So that's why it didn't come from this source, it didn't come from this source. Then I will see certain wells which show yellow color. Many cells will show yellow color. And what does that mean? It means that the gene expression of, let's say this is, let's say, cyclin gene. Okay, so my conclusion would be that cyclin gene was similarly expressed in both the cells. There was no change in expression. Okay, and then I will see certain wells which show a bright green color. So that means that that particular gene, let's say this is myosin gene, it is highly expressed in the normal cell, but in cancer cell there was very poor expression. So I can conclude from this particular thing that myosin gene is heavily downregulated in cancer cell as compared to normal cell. And similarly, I will see certain wells where I will see a dark red color, which will show that my particular gene of interest is highly expressed in cancer cell, while in normal cell, its expression was very low. And we will have a gradation of colors, and that's why when we have this chip, we have a particular scale that it ranges from, let's say, -3 to +15 or something. So that is done by the various softwares and all. So what is the advantage of this technique over the previous techniques is this, that at one time I can comment that of the 25,000 different genes which are present, let's say, in human cells, how many genes are up or down regulated in the cancer cells. So means simultaneous detection. But is microarray the only way you can do it? No, there are new techniques which help in measuring the transcript levels in various conditions, and that is next generation sequencing, which I will not cover in this particular course, but it is in more advanced courses where you can learn those techniques. But basically the idea is that you isolate total RNA from any particular cell or tissue of your interest, and then you send it for sequencing. Every transcript that is present in a cell is sequenced. And here also, from cancer cell also, you isolate total RNA and you get all the sequencing done. Then I will have to compare the number of sequencing reads between normal versus cancer cells. And there's a possibility, let's say for a particular gene, I find that in normal cell, there were 10 sequencing reads obtained for that particular transcript, while in cancer cell, I had seen 1,000 copies. So on basis of that, I can easily conclude that, okay, in cancer cells, my gene is, I don't know, I said 10 here and 1,000 here. So it means that it is 100-fold overexpressed in cancer cell as compared to normal cell. So that is just the whole idea behind sequencing method for understanding the transcriptome level changes. Coming to, so next topic is that how do we characterize the transcripts? At many points, you will have to know that what are the intron-exon boundaries. How do you decide that what are the intron-exon boundaries? or let's say you want to know, you have got a partial cDNA and you want to know the ends of the cDNA, what are the transcription start site, what is the end site, and so on. So with respect to that, I'll tell you few techniques. So if you have to map the intron exon boundary, it will be excellent, if you have cDNA in your hand and you have a genomic sequence in your hand, and you just compare the two. If you compare the two, you will obviously come to know that this region which is absent in cDNA but present in the genomic DNA is the region where intron exon boundary is there. But there are other methods also. So one is based on S1 nuclease. And if you remember in my early classes, I told you that this particular enzyme is the enzyme which cleaves single-stranded nucleic acid, be it single-stranded DNA or single-stranded RNA. Okay. So what I do is that I have my gene sequence and gene in hand, and I have my RNA in hand, and I hybridize the two, and then I treat it with S1 nuclease. So what will happen that obviously the region which was intron, because it did not find any counterpart in the RNA, it will loop. So because it will loop and it is single-stranded, S1 nuclease will degrade it, right? And then you remove you give it a alkali treatment, and you know that RNA is very unstable in alkaline condition. It will degrade, and what will we be left with is single-stranded DNA. And these small DNAs you can you can sequence, and you will come to know that okay, these are the these are the exonic sequences. And then when you compare it with the whole genome, the DNA sequence, then you can know that okay, these were the exonic sequence and the rest was the intronic sequence. So next is that if for a particular transcript, let's say you have this mRNA, and you want to know that what is its transcription start site. Okay, many times it could be that you've got through you made a cDNA library, and for whatever reason the cDNA that you got for a particular gene, it was incomplete. It didn't have the ends were not there. So how do you trace the ends of a cDNA? So for that there are three techniques that I'll be telling you. So one, and all these three are very simple techniques. It's only that I told you that as a molecular biologist, you just have to remember the function of various enzymes and all, and where you can put those into use. So anyway, one way is by S1 nuclease mapping. So what you do is that, let's say you know the region till here only and you want to further probe it. So you make the probe against that region towards the end of the transcript where you want to know the ends. If you want to know the end, let's say 3' end, so you will design a primer from here or probe from here, which goes up till here. So here I designed the probe, which extended for a longer stretch, and then I hybridize it with the mRNA, total mRNA. So what will happen that the probe will hybridize to the complete transcript which are present in the cell. Then I digest it with the S1 endonuclease, and obviously the region which is not, hybridized, which is present as single-stranded, it will be degraded. And then I denature it, and what I get is this particular stretch of the probe, the end of which represents the end of the transcript for sure, because in the cell, I'm assuming that whole transcript obviously was there. So this, you can run it alongside sequencing gel and you can know its size. And on the basis of its size, you can know that what is the end of this particular transcript that you are interested in. Similarly, another method is that you design a primer towards the end of the transcript for which you need to know the ends. And then you extend it with real-time PCR, with reverse transcription. So what will happen that only till the end of the transcript. It will extend it, and so it will go till the end of the transcript, and then you denature it, and then you run it on gel, and you run it on gel alongside the sequencing done with this primer uh, for each base. So once you run it alongside, the sequencing gel, then you can know its size, and then you can know that, okay, this is my transcription start site. Let's say on the sequencing gel, you come to know that, okay, this is, let's say, 100 base pair. So now I know that starting from here, I knew that the probe is from here, started from here till where is the 100 base pair, and that I will say that, okay, that is my transcription start site. Okay, and another method is rapid amplification of cDNA ends. This is race technique, RACE. Okay, so if you want to know the ends of the 5' end, then you will say it's a 5' race. If you are determining the 3' end, then you will say it's a 3' race. That's how it is. The nomenclature of this technique is. So let's say I want to know the 5' end of a particular transcript. What I do is that I make a primer from a regions which is, let's say, close. यहाँ पे इतनी दूर दिखा है, but I can also make it from here. Wherever I know, till where I know, for example, I know the end till here, I will design a primer here. Then what will I do? I will prepare cDNA. So it will make the cDNA till the end of the transcript. And after the end of the transcript, I then obviously means it will not go further. Then I denature it and I get this particular stretch. Now, how do I know what stretch it is? So what I do is that I add multiple A's on this side with the help of a terminal transferase. So if you remember, terminal transferase does not require any particular template for it to add the nucleotides. So you will add multiple A's here. And so now I know one primer here, and I can design oligo DT primer here. So with the help of a oligo DT primer and the primer which was kept here, I can perform PCR. Once I get the PCR, I can send this PCR product for sequencing, and it will tell me that, okay, what is the sequence from here till here. So the end of this sequence, just before A, is the sequence of the transcription start site here. Alternatively, what I can also do is that instead of adding A's here, I can also add any adapter here. So koi bhi adapter I add here, and I can design, because I have designed the adapter and I have ligated it. I can design a primer against that adapter, and I already have the primer against this region. I can again do the PCR, and then I can amplify it. Once I amplify it, I have two options. I can directly send this product for sequencing by purifying it from the gel, or means a purified product, I should send it. Or I can clone, because I have adapter here, and here also I designed the primer. I can clone it in my plasmid, any cloning vector, and make more of its copies, and then just send the plasmid for sequencing using certain primer sequences which are present on either side. They can be just universal primers for any particular cloning vector. So are these techniques clear? I mean, through these techniques, we have designed, we have characterized the transcript. How we have characterized, we could know that, okay, what are the intron exon boundaries. At the same time, if I wanted to know what are the ends of a transcript, I can know simply by designing a probe towards the end and performing S1 nuclease mapping, or designing a probe towards the end, and since it is a means labeled primer that I designed, I can go for running the final product on sequencing gel and knowing its sequence. Or alternatively, the through primer, the region that I've extended, the cDNA, I can add an adapter here, This primer I already had on this side, and I can amplify it with the help of Taq polymerase, which is the basis for this race technique, and send it for sequencing. So is it clear?
Yes, ma'am.
Okay. So next is that many times I would want to know, for example, I see that, let's say, normal versus cancer condition, I see that there is very high expression of um, any gene that I'm interested in, let's say some growth factor receptor, right? EGFR. So I see that it is very highly expressed there. But then I want to know that what is the reason for its high expression? Is it that the promoter is involved in its high expression, or is it simply the transcript stability is there and there is no influence of the promoter region? So to know how these genes are regulated, I need to do certain promoter based studies. And how do we do those studies? I'll just introduce you to those. So in your assignment section, I've already told you that for a particular gene that you're interested in, how you can go get the promoter region, how you can download the promoter region, right? In fact, you've also done the promoter reporter assays or no? So for your this assignment, I had asked you to, I think, download the promoter sequence and also mutate those, right?
Yes, ma'am.
है ना? प्रमोटर रिपोर्टर का कंस्ट्रक्ट तुम लोगों ने नहीं बनाया था. I'm not remembering actually.
We have just downloaded the 1 KB upstream region, and that we did. We were considering that as the promoter, that
ठीक
1
है,
KB upstream region.
ठीक है. So for promoter reporter assay also, you need to download the promoter sequence, and you need to design primers from the ends of the promoter region, and then you need to amplify that region and clone it in a vector which has a reporter gene. So what you have to do, you have to clone your promoter region upstream of a reporter gene. What that reporter gene could be, multiple reporter genes are there, so many which are written here, but the most common ones are luciferase based reporter gene, and GFP, and in case of plants, GUS. So these are various reporter genes, or beta galactosidase also, various reporter genes that you can use, and you clone your promoter here. Then what you do is that the construct that you have made, you transfect it into your cells. Now let's say you find that your gene of interest, let's say, what was that, BAG1 gene. That BAG1 gene, it is induced in response to heat shock. Let's say you had seen that at the transcript level by RT-PCR. Now you want to know whether that increase in transcript level is because of the increased activity at the promoter. So what I do is that I clone BAG1 promoter upstream of a reporter gene, and I transfect it into cells. So now I have two categories of cells. One in which in both of those I have transfected my clone. So in one, I gave it normal condition, and in another cell, or let's say cells which are growing in a well, I give a heat shock. So in response to heat shock, if I see, let's say, higher GFP activity, then I'm sure that in response to heat shock, there is some change in the promoter, or let's say some transcription factor, maybe it comes and binds to its promoter, which leads to induction in the transcription of that particular gene. Is this clear? So it is through promoter, means it is through the GFP that I've cloned here downstream of my promoter, that I can come to know that in response to heat shock, if I'm seeing higher GFP activity, it means that my promoter is heat inducible. So this will be my conclusion from the experiment that I've just done, right? And second thing is that if I do not see that there is increase in the GFP expression in response to heat treatment, then it means that the increase in BAG1 level that I was seeing in response to heat shock at the transcript level is not because of the promoter. It could be that it is some post transcriptional mechanisms which are involved in induction of the levels of that particular transcript. It could be that under heat shock, that transcript got more stable as compared to under normal conditions. Okay, so this is how you have to make your conclusions. Okay, so now coming to that, the same thing, that is promoter luciferase assays or promoter reporter assays. Okay, so how do you? So there is something also called as promoter deletion analysis, where in the whole promoter that you have cloned, you want to specify. Let's say in the previous case that I had told, that let's say you cloned whole 1 or 2 kb promoter, but within that 2 kb promoter, let's say you saw that in response to heat shock there was very high activity, but now you want to sort it down to that which are the specific region within the promoter which are responsible for heat inducibility. You want to shorten the region. So how do you do it? By deletion analysis. So you make deletion step by step in the promoter. Let's say this was the whole promoter. I think it's around 1.5 or 8 kb, whatever. So this is, let's say, the whole promoter, and then you make its deletions, and then you see that maybe till here I see very high GFP activity, like in the example I told you before. But when I deleted it further, then that activity of heat inducibility completely went. I did not see an induction in GFP. So what will I conclude? That the region for heat inducibility lies between this and this region. Is this clear? I'll come to other examples which are shown here, but I wanted to relate it with the previous example that I told you. Is this clear why you want to do deletion analysis step by step?
Yes, ma'am.
Yes, ma'am.
ठीक है, तो इसमें यही करते हैं. You know the region here, you design one set of primers from here till here. तो तुम्हारा whole promoter clone हो गया. Then you again design primers from this to this, then further down, then means further down. So that way you can amplify the promoter of various lengths and clone all of these upstream of a reporter gene. And then you come to the conclusion. The example which is shown here is for the region where they are trying to look at which region is responsible for its expression in the, I think these are leaves, in the leaf region. Okay, so they know that in this 4 and 5 construct, which is these two, ठीक है, इतने छोटा करने के means in these two constructs they did not see any expression of the reporter in the leaf region. So there was no expression at all. Rather, I would say maybe probably this is the minimal promoter for it to have any expression. This is the minimal promoter likely, because before that you don't see any expression only, and then when you increase it further, then you see a very strong expression here. So likely its inducibility region lies here in this region. So that's how you have to come to sort down that which region is important. In some cases you do this in silico analysis of the promoter to look for certain tissue specific region, or some transcription factor binding sites which are present here. So let's say I come down to that, okay, probably a transcription factor which is involved in, let's say, seed specific expression, likely it binds here. I want to be sure that that region is very important for it to be expressed in seed. So what I do is, one, I clone the whole promoter upstream of reporter, and in one case what I do is I remove that particular region. How I can do is by deletion analysis. I need to find certain enzyme sites which are flanking this particular region and then just religate it. So now this particular stretch, if now it does not show any seed specific expression, I can be sure that the region for seed specific expression lies here. I can conclude that. So because deletion of this leads to expression of that gene in all tissues and not specifically in seed. Okay. So now coming to the last two techniques, which is that I know, let's say in this case, there is some region which is important for its expression in seed, right? Now I want to know whether any protein binds here or not. Is it bound by any protein or not? Or let's say if I'm interested, I know that transcription factor, let's say LEF1 transcription factor binds here, and I want to confirm that it really binds here or not. So for that I go for EMSA analysis. As the name says, it is electrophoretic mobility shift assay. It means that we have to perform electrophoresis of DNA fragments we are interested in, and we look for the shift in the mobility. Why we are assuming that there will be a shift in the mobility? Because if the DNA is bound to a protein, then we will see that its mobility on a particular agarose gel or page gel, it will be very much hindered. So that's why we will see that it is we will see the shift in the mobility of the same DNA fragment when it is bound with a protein. If it is bound with multiple proteins, we can see multiple bands also, because there can be some DNA fragments where only one DNA binds, where one protein binds or two proteins bind, and so on. So this is the basis behind EMSA assay. So what you do is that you the DNA, let's say in this case, this particular stretch. Okay, you take this particular stretch and you label it with radioactivity, or you can also use, now there are kits for making fluorescent probes as well. Okay, so when you run it, simply in one lane, you run just that DNA, which has been labeled. And what you also do is that you incubate. Don't look at this figure as of now. I'm just telling you this figure. So then what you do is that you incubate your DNA with the protein with increasing amounts of the protein. ठीक है, एक ट्यूब में तुमने डीएनए लिया और उसमें प्रोटीन डाला. In another tube you added more DNA. In another tube you added further more DNA, and then you run all these samples on the well, on the gel. So what you will see that in this case, when comparatively lower protein quantity is there, and let's say your protein is binding to the DNA, then you will see a mobility shift. If let's say you are, probably your hypothesis was wrong, your protein is not binding to that DNA, what you will see, you will not see any mobility shift. It will, all the DNA will be present here. Okay, so what you then do is you increase the protein quantity, and with that increased quantity incubated sample, you run it on gel. Then you will see that this band is disappearing, and you will see all the band here, all the bound DNA with the protein is present here. Okay, now to know that whether this binding is really specific or not, I provide one option is that I provide my this DNA probe, which is a DNA fragment which is not labeled in much excess as compared to this. Same DNA fragment in much more quantity, but without the labeling. So what will happen that in the tube in which I've added the competitor, apart from my probe and the protein, what will happen, because the competitor, which is just not labeled DNA, it is in much more quantity as compared to your DNA probe, I will see that it will bind to the protein, but because it is not labeled, I will not see it band, and still I will see that my probe is present here. So there are certain experiments that you do to ensure that your binding is specific. So in this example, so this is the real gel, this is the, just a cartoon. And here I wanted to know, so this is, I think, some figure again taken from Google. Just to tell you, so this is some, I think, Epstein-Barr virus DNA or whatever. So this is the DNA, and they wanted to check its binding with the protein EBNA protein. Okay, so this is just the DNA which is labeled. Here it is incubated with the Epstein-Barr virus extract. And I do see, or in some cases, if you're very sure that which protein binds to it, you take the purified protein. Okay, so two things that you would require will be the DNA fragment which is labeled and a purified protein if you have it. If you have no idea that which protein binds to my gene of interest or DNA of interest, then you can just take the whole lysate and bind to it and incubate it with that. So in this case, I had that idea. So anyway, so I see the mobility shift. And then when I am adding unlabeled eBNA DNA, like I told you in this case, then you see that this band is disappearing and we see band here only. Okay, so that it is competitive uh, competition basically. Then in the last case, what I've done is that instead of adding the unlabeled eBNA DNA, like I told you here, I take some totally non-specific DNA. In this case, oct1 DNA. ठीक है? Means ideally इसको competition नहीं करना चाहिए. So it's like a negative control. So when I incubated it with totally unspecific DNA, then I did not see this band disappearing. Okay, so this is by having various controls, I can come to a conclusion that the protein that I'm interested in is specifically bound to my gene of interest. So these are some sophisticated molecular biology experiments that you have to do when you are working in a lab and interested in a particular DNA protein interaction, or in the you're working on regulation of a particular gene, how it is getting regulated. So the last technique that I want to tell you will give you the output that, see, in this case, I just come to know that in this particular stretch of DNA, it can be, 500 base pair or 200 base pair stretch, that yes, a protein is definitely binding here. But how will I know that where in this whole stretch it is binding? Through this experiment, I just come to know that it is binding. But what is its position? How do I know that? By a technique called as DNA footprinting assay. Okay? We see for the footprints of a protein bound to a DNA fragment. So again, you take that DNA fragment that you are interested in, and you know that a protein is binding there. Okay? Then you label it on any one end and you incubate that DNA with your protein of interest. Okay, so we are assuming that in the tube now, where you have this labeled DNA and protein, the protein gets bound to a particular place in the whole this stretch. Then you treat it with DNAs. So, and you give a very mild treatment with the DNAs, such a way that it makes breaks at various places where it can. The point is that the region which is bound with the protein, it will not be digested by DNAs, because it will be protected from digestion. So what you will get a fragment this size, 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 and then directly this size from here till here. Then this size, this size, this size, this size, and so on. right? So because of this thing, you can, and then you run it on gel. So what you will see is the footprint of that DNA. So you run it on gel and you figure out that the bands which are missing here are the region where the protein is actually bound. So here also what you do is that you increase the protein concentration. So you have 4 different tubes. In one, I do not add the protein and I perform the DNase assay. And in second, I add little bit of protein. In third, I add more protein, and in four, I add all the more amount of protein. So then you will see these disappearing bands in between. So these bands will tell you the position of the protein where it exactly binds in the whole stretch of DNA. So that is all for today's class. Do you have any questions?
Ma'am, while we were doing RNA isolation, you said that we need to be very careful that it does not degrade, and we need to even keep our mouths away from the tube. So can we do it in laminar airflow then?
Yeah, you can do it, but I the only thing is that in a laminar airflow, do you really ensure that the protein are not there? It is the airflow, but can you still ensure that the enzyme is not there in the air? So
Uh, yes.
the best thing is that you try to keep these tubes open for limited amount of time. You should just say, sometimes we just get casual that you keep the tube open, or you are, talking in front of the tube and all, the fans are on and all, but it is prescribed that you should do minimal activity with your RNA sample so that it is preserved and doesn't get degraded. In many cases, sometimes people also add RNase inhibitor in the RNA, let's say when you are doing cDNA preparation and all, so sometimes you also get RNase inhibitor for certain sophisticated RNA techniques.
Okay, ma'am. And ma'am, in the major exam, because in many of our courses it is there that the syllabus of minor was not included in the major. Will it be same
Yeah,
for
yeah,
this
yeah,
course as well?
yeah. Yes, yes, yes. So
Oh,
in this course, after minor, whatever has been done will be part of the major exam.
okay, ma'am. And nothing from the minor one.
Nothing from the minor one.
Thank you.
Okay. But main thing, see, my thing was that through assignments and practicals, you understand that how do you isolate DNA sequence, how do you design primers, do you design siRNA, shRNA. ये चीज please बिल्कुल मत भूलना, because any molecular biology lab you go to, you will need these basic experiments that are required.
Yes, ma'am.
ठीक है. Okay. Anything else? I will anyway send the email for uploading the assignment on the Moodle rather than sending it as an email, and do it maximum by tomorrow, please. Okay? Those who have not already done it. I think majority are the BTech, MTech students who have sent it as an email. Okay, guys. If there are no more questions, then bye bye. Have a good day.
Goodbye, ma'am. Thank you.
Bye
Thank
bye.
you, ma'am.
Yeah.
Thank you, ma'am. Bye.